system, excuse me, they have, as I just was discussing this a little bit previously, like the alimentary canal or GI tract. The alimentary canal consists of mouth, from the mouth, pharynx, esophagus, not larynx. Larynx and trachea goes to the respiratory system. Here, esophagus, stomach, small intestine, and large intestine. That's the GI tract. That's the alimentary canal. Mouth, pharynx, stomach, small intestine, large intestine. And accessory organs right here, you have teeth, tongue, and salivary glands right here. And in the abdomen, you have liver, gallbladder, and pancreas. So these are the accessory organs. They help the function that digestive tract or the gastrointestinal tract want to achieve. So what is they want to achieve? They want to ingest the food, meaning take the food inside. They want to digest the food. They break the food chemically, and then they will absorb the material that body can handle, body needs them, and the remaining they can excrete the food. So that's basically the main activity of the digestive system. Ingestion, like literally take the food in the mouth. Now, and then once they accept the food in the mouth, they do propulsion, movement of the food, because food cannot walk. So our body needs to help them how the food can move. So those are the help of the muscular activity. So I'm going to come back right there. So those muscular activity include peristalsis, and activity includes peristalsis and segmentation. So peristalsis happen in the esophagus area, and segmentation happens like pharynx and esophagus, that's peristalsis, and segmentation when it's taking place in the intestine. Bottom line is same. The both movement is very similar scientifically. So the difference is this, like the movement is kind of like the way you take out toothpaste from a toothpaste tube. So how do you take out toothpaste in the morning? You just press on the back so the toothpaste comes out. So you keep pressing on the back of a toothpaste tube so that you can force the material, the paste inside them towards the opening. Same thing here. They keep contracting the muscle on the back of the food, and so that food literally can walk. So that's peristalsis. Peristalsis in this case, like one bolus goes there. So from mouth, when you swallow, that one bolus goes to the stomach. Then you swallow again, there's another one goes to the stomach, like a one at a time. But in small intestine, like a lot of bolus are moving together. So the same technique, the technique is same, they just segmented it. That's why the new name is segmentation. So this is in intestine, and this is in the peristalsis is in pharynx and esophagus area mainly. Okay. So ingestion, when you take the food, propulsion, the movement, okay, mechanical digestion. So the digestion can be divided into two halves, mechanical and chemical. Mechanical is you are not changing the chemistry of the molecule. So let's say you take a French fries, which is potato, starch, polysaccharide. So the chemistry is you are in your mouth, you have the starch or a piece of French fries, and then you are chewing them, making a smaller piece of starch, like a smaller potato. You are not chemically digesting them. Then when you add enzyme to it, then you are chemically changing the starch. So then starch becomes smaller molecule, which eventually turns into glucose. So that is a chemical digestion, which requires enzyme. And those accessory organs, along with your digestive tract, secrete many enzymes. Then you absorb. So glucose, yeah, we're going to absorb, but cellulose, the fiber, I can't absorb. Human body doesn't want to absorb those fiber, but they're required in the food because they help in the bowel movement, but we don't want to absorb it. We defecate them. So we eliminate those indigestible solid waste. So we throw them out. So ingestion, you take the food in, you move the food propulsion, you digest them mechanically or chemically, like mechanically and chemically, I should say. And then you absorb them, the one that you need, then you don't need, you release them. That's basically your whole gastrointestinal activity. Now, first thing first, so we got to know that gastrointestinal tract or GI tract, how they look like microscopically. There are 4 layers from the lumen, the opening, to the outside. There are 4 layers from the lumen is the opening. From the lumen, there are 4 layers. So innermost layer is called mucosa. The next layer is called submucosa, followed by muscularis externa and serosa. So if you look into this structure right here, we can see this is the lumen right there. That's the lumen, that's the opening. And this is layer number 1, mucosa. So from the pink color to the red color right there, that's a mucosa. Then this is submucosa. Then that's pinkish color, that's muscularis externa. And then right here, the yellowish color, that thing is called serosa. So we need to know what they made of. Mucosa has 3 things in it. The mucosa itself has 3 things in it. The epithelial cells, so that's epithelium. Then next is the basement membrane for epithelial cells, because we know all epithelial cells have a basement membrane. That's one of the characteristics of epithelial cells. Okay, so that's a basement membrane. That's a fancy name called lamina propria. That's the name of that basement membrane. And finally, they have a very tiny layer of muscles, smooth muscle, that called muscularis mucosa. They have a little bit of muscle layer here as well. That's called muscularis mucosa. Then submucosa, that's full of connective tissue. Lots of blood vessels are there, nerves are there. They innervate the submucosal layer, lots of glands are there. And then comes muscularis externa. That's also a smooth muscle layer. And finally, serosa with a little bit of epithelia covered by connective tissue. A little bit of epithelia covered by connective tissue. So that's histology, meaning how the tissue looks like. Now, one more time, in the mucosa layer, there are 3 things: epithelial layer, lamina propria, the basement membrane, and in the basement membrane, there's some glands are there. And then finally, the smooth muscle layer, the tiny smooth muscle layer, a little bit, not too much. Now, question is, what kind of epithelia is there? Now, according to the arrangement, if you remember, epithelium are like 2 types, simple and stratified. Simple means 1 layer, stratified, multiple layers. According to the shape, they are either squamous or columnar or cuboidal. Your gastrointestinal tract deals with 2 of them, squamous and columnar, not the cuboidal one. And they also have some simple and some stratified area. Now, which one should be simple, which one should be stratified? That depends on your simple understanding. Stratified, obviously, where you need to deal with rough material, because multiple layer of the epithelia will prevent any kind of damage of the tract. And simple layer is where you do not need to deal with any rough material, so that even if they are dealing directly with the food material, they're not going to cause more damage to the mucosa. So now if you think like what area of the body in the gastrointestinal tract, starting with mouth to the anus, you deal with rough material. So when you take a bite of like a sandwich, that's the area dealing with the rough material. So your mouth, your pharynx, your esophagus, this is the area the food material is not softened yet. So up to esophagus, or the junction of the stomach, there your epithelia will be stratified squamous. Then once it reaches into the stomach, small intestine, and most part of the large intestine, they're dealing with the soft material. It's like mostly liquid because body secrete a lot of fluid into it. So it's like a soupy material, squishy, soupy material. So they are all simple. Then the last part of the large intestine, which is the rectal canal and anus, but because large intestine, we're going to learn later on, they're going to absorb a lot of water back into the body. So the material become rougher again. So the rectal canal and anus, they have the stratified epithelia. That is how you can understand and then memorize that exactly where you have stratified and where you have simple epithelia. Wherever there is simple columnar cells, stratified squamous cells. Excuse me. Muscularis mucosa, that's a smooth muscle layer. Now, this smooth muscle layer is also same as here. That's also smooth muscle layer. There are two ways the smooth muscles are arranged in this case. One is circular, meaning they're going around the tract. So if this is a tract, they're going this way. So that's a circular muscles. And the other one, the muscles is running lengthwise. So they call longitudinal muscles. So in this particular example, right here, this is a circular muscle. They're going circle around this lumen. And this piece of muscle that's going lengthwise, the longitudinal. Circular is the innermost, longitudinal is the outermost. So this is circular and then outside is longitudinal. That's how the muscle. That is true for both muscularis mucosa and muscularis externa. Both, although tiny, they still have two layers. Innermost circular, outermost longitudinal. Innermost circular, outermost longitudinal. Both the cases. Muscularis mucosa and muscularis externa. Obviously, in the middle, they have some mucosa full of gland and connective tissue. Now, one extra point here for muscularis externa, in the stomach. So from mouth to anus, as I said, all four layers, all this serosa from the outside, like serosa, muscularis externa, submucosa, mucosa. So same, very similar. We now know the difference in mucosa. And there is one difference in muscularis externa also. In case of stomach, I'm going to mention one more time in stomach again. In case of stomach, there are three layers, not two. So everywhere else, there are circular muscle, longitudinal muscle, smooth muscle. But there is oblique, circular, and longitudinal. There is one oblique direction also. The stomach is one of the most strongest in this case they can temporarily store a lot of food and they need to turn and churn to create a lot of gastric motility that helps digesting the food material. That's why sometimes your stomach growl when they have like a lot of liquid in it. So remember that way, they have three layers of smooth muscle: oblique, circular, longitudinal. Oblique, circular, longitudinal. And finally, serosa, their epithelial layer, and then on top of them, they have a connective tissue covering to protect them. Most places are like that, but in esophagus, particularly in esophagus, and even in the pharynx area, that outside connective tissue and epithelial combination, that serosa is much stronger because they're running through the cartilage and bony area of the body, not in the abdomen. Abdomen is already everything soft, but here is like a bony area. So that esophagus is literally running back of the trachea, which is made of cartilage, and in front of the vertebral column, which is made of bone. So they need a little bit more tougher. So their serosa become modified. We call them adventitia. So modified serosa. It's nothing. It's still fourth layer. It's just much more tougher. So whatever I discuss about the mucosa should be right here. Whatever I discuss about the, this is again, the adventitia of the serosa, that's what is mentioned. Adventitia in the esophagus, that's the circular and longitudinal, muscularis externa, submucosa, whatever I said should be there. These are the different organs one more time. So for the anatomy purpose, that's mouth. From mouth, this is the pharynx area, the long pipeline. Esophagus is connected to the huge J-shaped organ called stomach. Then there is a convoluted pipeline we call small intestine, and finally the large intestine goes to the anus. So the small intestine and large intestine, they call small, but actually they're very long. They're called small because the diameter is small. They call large. This one is a large because the diameter is large. Okay, but the small intestine is actually very long. And then this is the large intestine. Right here, we have the salivary glands, 3 pairs, and tongue and the teeth. Those are the accessory structure. Right here, we have gallbladder, liver, and pancreas on the background of stomach. So those are the 3 accessory structures. So 6 total accessory structures, and then in the whole tube, starting from the mouth to the anus, that's digestive system. So for the anatomical purpose, like when you're doing the lab, stuff like that, you got to memorize all of these, like which one is what, and then detail about this. So let's start with this top. So again, the top is like mouth, then pharynx, then esophagus. That is how we are moving for the tract. And when you study mouth, it is better to study teeth, tongue, and salivary gland at the same time, because together they have a very similar function. Okay, so so mouth, obviously, they're bounded by your lip, cheek, palate, and tongue, and the mouth known as oral cavity. Now, the first thing, in order to understand the function of as a whole, we need to know what mouth function is, like what mouth does. So basically, mouth accomplish few very simple functions. The first one is like they house the ingested food. So when you ingest in the mouth. That's the first thing. Now, once you take the food in, you taste them. You taste them. That's why we don't eat pens or peppers, because they're not so-called tasty. And then our brain figure out, okay, that's not my food. So the taste bud on the tongue helps them to do it. So you taste them, you analyze them based on our experience. That is, again, you are not going to eat the pen because we analyze that this is not a food. So tasting and analysis takes place in the mouth. Once we do that, then we started chewing them. So that's called mastication. So the muscles and the tongue and the teeth, they play a role in it, and also salivary gland, because they pour saliva in it, so they make it soften, moist. Okay, so then you have the moist food, and then because if the food is dry, it's difficult to swallow. So that's the next part. Once it is chewing, so making a mechanical digestion is happening. So you're breaking down the food small, and then you swallow. So the swallowing, the word is called deglutition, and I do not have a separate slide on that, but you should listen carefully this one. So swallowing is the method or phenomenon is called deglutition. Deglutition means swallowing. So when you swallow, it has 3 phases. So buccal phase, buccal cavity means mouth cavity. So buccal phase. Then pharyngeal phase, meaning when it is near the pharynx. And then finally esophageal phase, meaning the food material goes into the esophagus. So 3 steps or 3 phases of deglutition or swallowing. So buccal phase. So once your chewing is over, your buccal phase with your tongue folded backwards, the palate goes up, you're pushing the bolus inside. So the bolus is moving from here, right this area. So that basically right over here, that's called buccal phase. When it's in the pharynx, we're calling them pharyngeal phase. And finally, when it's in the esophagus, the peristalsis already began. That's called esophageal phase. So out of this, like buccal phase is the voluntary phase, meaning that's voluntarily controlled by you. All the skeletal muscles are engaged. They are helping you to decide whether you are going to swallow or you're going to spit it out. But once it is there, then it's involuntary. You don't have to worry about this. Then the esophagus and pharynx, they will take care of it, take it to the stomach, and so on. So that's in general the function. Now, a little bit additional role in mouth. One extra feature is when they're breaking down, not only they are performing mechanical digestion, they also participate in a little bit of chemical digestion. So that's why it's called partial digestion of food taking place. What kind of food? Particularly carbohydrate. The reason in salivary gland, in saliva, the secretion of the salivary gland, saliva contain an enzyme which can digest complex carbohydrate, not simple carbohydrate, complex carbohydrate like polysaccharides. So they can digest starch, they can digest a little bit of glycogen, so they can digest a complex carbohydrate molecule, not glucose, not sucrose. Those are simple like monosaccharide and disaccharide. We are not going to digest them in the mouth. So mouth participate in partial digestion of food. So we can say carbohydrate digestion begins at the mouth. Protein, nothing happened to the protein because there is no enzyme in the protein that can help it. Although, however, mouth release, like tongue release, a lipase. We call that lingual lipase, but that does not efficiently work in the mouth. So some book will mention that the tongue release a lipase, breaking down enzyme that breaks down lipid, but that doesn't happen efficiently because lipid breakdown required a phenomena which I'm going to discuss later on. It's called emulsification, and that takes place in the small intestine. So before the fat is emulsified, we don't digest them efficiently. So we are going to ignore that part. So in the mouth, one digestion happens, and that is partial digestion of the carbohydrate. So that means that the enzyme in the mouth, namely salivary amylase, amylose is the other name of starch. So amylase, or some book will call alpha amylase or tyline. These are all other name of the same enzyme. They help digesting complex carbohydrate. Complex, that's a keyword. Complex carbohydrate, meaning polysaccharide. So that's basically the function of the mouth and teeth, tongue, salivary glands all together. But individual information a little bit. This palate, the structure of the mouth, we were reading, tongue. So a little extra piece of individual, because tongue is a very common mistake. People think it's a smooth muscle. No, it is a skeletal muscle, because you learn the skeletal muscle is the voluntary muscle. So the question yourself, you have to ask yourself is, can I move my tongue voluntarily? The answer is yes. So this is a skeletal muscle. And there is a membrane called lingual frenulum right here underneath the tongue that is securing the tongue to the floor of the mouth. So tongue is filling the floor of the mouth and movable structure, maybe the skeletal muscle, and lingual frenulum, the membrane holding securing the tongue. So what they're doing, they're helping you to mixing the food with the salivary glands material, which is saliva, and then helping you to chew the food, and then helping you to swallowing the food. On top of that, they're helping you to analyze and testing the food and in the speech, which part of the respiratory system. Salivary glands, 3 pairs of salivary glands. First thing first, you need to know their name. Parotid gland, submandibular gland, and sublingual. All are paired to 2 parotid glands right here underneath the ear, right over here. Submandibular underneath the mandible, and sublingual underneath the tongue. So we have 6 of them, 3 pairs of salivary gland. Together, their secretion is called saliva. Together, their secretion is called saliva. So saliva, again, same thing, we already know it, cleanses the mouth, moisten and dissolve the food material, bolus formation, they help in bolus formation, and they contain salivary amylase that break down starch, complex carbohydrate. So these are the location, and all 6 glands, meaning all 3 pairs of glands, are exocrine gland, meaning the gland with the duct, not endocrine, not hormonal gland. These are secreting saliva, but they are secreting through the pipeline. So parotid gland, they have a pipeline attached called parotid duct. Submandibular gland they have a pipeline attached called submandibular duct. The sublingual gland, they have pipeline attached. Those are called sublingual duct. So all these glands, all the 6 of them, they are exocrine gland. That means they have a pipeline attached. So whenever they secrete, it fills your mouth. So saliva does not like come up right here or here. They just go into your mouth because they have a pipeline attached. They are exocrine gland. So for anatomy purpose, you must know their location and the name of the gland and the duct. What they say, what saliva, what they have, they're like 99% water, obviously. pH is 7. We can go with 7. pH is 7 means neutral, like 6.75 to 7. So we'll go with 7. pH 7. They have a lot of electrolytes: sodium, potassium, chlorine, phosphate, bicarb, a lot of electrolytes. The one main thing they have is salivary amylase or alpha amylase. That's the enzyme. That's the digestive enzyme. What they can do, they can digest. They can digest the starch. So they can only work on a complex carbohydrate. They also have a little bit of protein, mucin, which react with water, become mucus. So they create a slippery thing for the food material. So the food material can be swallowed easily. Lysozyme, the enzyme that are responsible in a little bit defense mechanism along with defense and IgA. So these are a little bit lysozyme, defensin, and IgA. This is an antibody. These are something to do with the immune system. So that means they are defending your body as well. They produce a liter and a half per day. So that's a lot of saliva. You also need saliva, like not only because of swallowing, but for speech. So when I'm talking, you need saliva, and sometimes you swallow this to make sure that it helps you in the speaking ability as well. Pharynx, so that's mouth. So when you study mouth, you study function of the mouth and then function of the tongue and salivary glands. And at the same token, you got to know the teeth. So I probably don't have a slide on teeth. Yes, I don't have a slide on teeth, but to make sure teeth, what do you need to know about the teeth? 32 of them, 16 on the upper jaw, 16 of the lower jaw. If you divide into 4 quarters, 8, 8, 8, 8, and we talked, so let's one more time. So right here, if you divide this, so on the top 16, on the bottom 16, so 16 on the bottom, 16 on the top. If you divide into 4 quarters, 8 here, 8 here, 8 here, and 8 here. All of them, they have 2 incisor, 1 canine, 2 premolar, 3 molars. 2 incisor, 1 canine, 2 premolar, 3 molars. The premolars and molars, they grind the food. Canine, they tear the food, help you tearing the food, and this is cutting the food. So together, all of them, along with the saliva, they chew the food. Okay, they chew. That's called mastication. So they help in mechanical digestion, and that salivary amylase, that enzyme, perform a little bit of partial digestion or chemical digestion as well. So this material, they're very hard material too. So they are basically in a socket. So the tooth is sitting in a socket. So tooth is literally, this is a socket, this is the tooth. So they're sitting right here in the top. So in studying a structure, individual structure of the tooth, we are going to go that detail later on, not right now. Okay. Pharynx, we studied that in the respiratory system already. Pharynx has 3 parts: nasopharynx, oropharynx, laryngopharynx. Nasopharynx is something to do with the air, but oro and laryngopharynx both carry air and the food, both material. The naso only carry air, but oropharynx and laryngopharynx, they carry air and the food, both. Food goes to the esophagus, air goes to the trachea. If air goes to the esophagus, it's not a problem, but food cannot go to the trachea. There's a part of the larynx called epiglottis that act like a chute. So when you're swallowing, they close this up and the food material slide down into the esophagus without going into the trachea. Esophagus is like a excuse me, foot-long pipe that just carrying food. So they start from the pharynx, like the bottom part of the pharynx, which is laryngopharynx, and they go directly to the stomach junction. That's called esophagus stomach junction, or sometimes known as cardiac sphincter. There's a sphincter bottom there. So at the bottom of the esophagus, there's a sphincter. Sphincter means like a valve, muscular valve, like a rubber band right there. So the acid in the stomach cannot just go up in the esophagus. So the sphincter open and the food material goes there and sphincter get close. That's area, that sphincter is actually called cardiac sphincter based on the junction called like a cardiac orifice. Orifice means opening. Okay, that's basically esophageal and stomach or gastroesophageal junction, that bottom part. Esophagus does not participate in any kind of digestion nor any kind of absorption. Mouth still have a little bit of digestion factor, but esophagus does not participate, and also pharynx does not participate in any kind of digestion or any kind of absorption. They just transport the food, peristalsis. So they take the food from the top, from the pharynx, boom, goes to the stomach. That's it. That's their function. They just transport the bolus using peristalsis. Okay, we learned this. Now the stomach. So stomach is a huge, like a letter J-shaped organ. So they divide into four parts: cardia, fundus, body, and pylorus. So this portion is the cardia. That's what the esophagus is. That's what the sphincter is. So that sphincter is called cardiac sphincter or gastroesophageal sphincter. The sphincter, like a valve between the esophagus and the stomach. Stomach is referred to as a gastro as well. So this region we call cardia. This upside down, like U, that area we call fundus. Fundus, upside down U, that kind of structure. And then entire portion is called body of stomach. And final portion right here, we call them pylorus. Right there, there is another sphincter, another valve in the bottom, we call pyloric sphincter. That sphincter between the small intestine, which the first part of the small intestine is called duodenum. So between the small intestine and the stomach. So stomach is a huge J-shaped organ divided into four portions: cardia, fundus, body of the stomach, and pylorus, the bottom portion. At the cardia, we have a sphincter called cardiac sphincter or gastroesophageal sphincter. In the pylorus, we have a sphincter in the bottom. We call that pyloric sphincter. These two sphincters is like a rubber band, boom, like a tight. So they're holding tight. Why? Because inside the stomach, the environment is extremely acidic. Inside the stomach, the pH is close to like 1.5 or like a 2.0. So 1.5 to 2, that's the pH. And we remember a little bit of chemistry. pH 7 is neutral, which is here in the mouth. pH below 7 is acidic. More you go towards 0, the stronger the acid. So below 7, like 6.75 is acidic. Now you're talking about pH 2. It's a very strong acid. More than 7 is alkaline or basic. The small intestine a little bit this way. Mouth is right here. Stomach is acidic all the way here. Because the enzyme, the particular enzyme that I'm going to mention next, that enzyme, their optimum pH is pH 2. Okay. That enzyme coming up, pepsin, the name of that enzyme is pepsin, which digests protein. So we can say protein digestion can take place in the stomach, or protein digestion begins at the stomach. Before we go there, a little bit more anatomy. There are 3 layers in the muscle. Muscular is external, that was the exception mentioned before already. Oblique layer, circular layer, longitudinal layer. So that's the oblique layer, then regular, circular, and the lengthwise. So this goes all the way here, this goes all the way here, lengthwise. That's called longitudinal, okay? And not only that, in the mucosa area, where we have like simple columnar, that mucosa produce lots of ups and downs, like this, a lot of ups and downs. Those ups and downs created like a ridge-like structure. Those are called rugae of mucosa. Those ups and down structure of the mucosa helps material to mix up very well. It is kind of like, you know, when you're using the clothes dryer, So you put your washed clothes in the dryer. If you look at the dryer, they have kind of like a ridges inside. It's not a plain simple drum. It has ridges. Why? If you see next time that the clothing are moving, those ridge helps them to move better. That's exactly the similar function here. So those ridges helps food to get mixed with the stomach juices really well. Once again, the stomach is divided into 4 parts: cardia, fundus, body, and pylorus. 2 sphincter, cardiac sphincter right there, pyloric sphincter here. 3 muscularis external layer. In the muscularis external, 3 smooth layer, oblique, circular, longitudinal. Ups and downs of the mucosa is called rugae. Digestion of the stomach. So that's the very important part. Like, what is happening in the stomach? Stomach involves a lot. First of all, they're temporarily storage center for the food. So stomach temporarily store food. That's the number one thing. Like when you say eat, like that's why your stomach bulges a little bit when you eat a lot of food, because they are temporarily stored. Now, in the stomach, they're also participating in digestion, not like esophagus. Esophagus just carry the food. Stomach, when they're holding it, they also digest them physically and chemically, both. Physically means stomach is churning, so they're mixing. So obviously some kind of physical breakdown will take place. And then chemical. What kind of chemically? They secrete an enzyme called pepsinogen, that's an inactive form. They become pepsin, that pepsin digests protein. So we can say protein digestion begins at the stomach. That's it, that's one digestion. And the remaining is all in small intestine. So they create a chyme, the bolus. The food material is here. Once you swallow them, we call that a bolus, food bolus. When it mixes with the stomach acid and juices, we call them chyme. That chyme, the liquid material that goes into the small intestine for further digestion. Okay. So enzymatically digest proteins with the enzyme called pepsin. That's the enzyme. And stomach also secrete, apart from hormone, they also secrete 2 very important things. One is an intrinsic factor. The second is hydrochloric acid. That's why the pH is 2. So intrinsic factor, that factor, I think you learn in the blood anemia chapter, if that factor helps vitamin B12 to get absorbed. If you cannot absorb vitamin B12, you have a pernicious anemia. Vitamin B12 deficiency, we learn in anemia that they cause pernicious anemia. That's the intrinsic factor secreted by the stomach. If they don't secrete that intrinsic factor, then you cannot absorb vitamin B12. If you cannot absorb vitamin B12, you are going to end up in anemia. Now, if we go even detail, like what is the microscopic structure? Like what kind of cell you have? Let me see. I will take it right from here. You have. So not only not only epithelial cells that you have in the epithelial cells lining. Remember, mucosa is epithelial, so they are simple columnar cells. In that cells, you have some goblet cells. So that goblet cells produce alkaline mucus. So remember, stomach is very highly acidic. So they produce a little bit alkaline mucus to keep acting as a buffer. So the cells are safe. They do not get damaged from that strong acid. That is why you have 2 sphincter. Because if that acid leaked out to the duodenum, to the small intestine, or that acid leaked out to the esophagus, you know the burning sensation. Sometimes if you have indigestion, you're throwing up, the acid comes up and you know that acid literally burn here because that's very strong. So they can cause a lot of damage, eventually cancer. Initially ulcer, we call like esophageal ulcer or duodenal ulcer, people suffer from, and that ulcer, if not treated properly, cause a lot of acid burn. So that's what we call like burning, like, you have like heartburn, so called. That's because of that acid secretion. That's why those 2 sphincter has to be tightly in place. So but how does stomach himself protect it from those acids? Because of these goblet cells. So they create a layer with the bicarbonate in it. So this mucus layer, they traps bicarbonate-rich fluid underneath. And the bicarbonate is alkaline in this case. So they protect themselves from that by that cells. And now obviously they have many other cells secrete many other things. So these are the main two cells. Apart from the mucous cells, there are main two cells in the stomach. They secrete what they call parietal cells and chief cells. So parietal cells, they produce hydrochloric acid and intrinsic factor. They produce both hydrochloric acid and intrinsic factor. Intrinsic factor function we already know. Intrinsic factor helps absorbing vitamin B12. That's their function. What HCl does, this HCl, hydrochloric acid, they activate pepsinogen. Now the question is, what is pepsinogen? Chief cells, the other cells in the stomach, they produce an inactive form of pepsin called pepsinogen. That pepsin is inactive. Pepsinogen, ogen, inactive enzyme. This HCl cut down this, convert into pepsin. So pepsinogen is activated to pepsin by HCl. Now what pepsin does? Pepsin digests proteins. Pepsin digests proteins. So protein digestion begins in the stomach. So one more time, parietal cells secrete HCl and intrinsic factor. Intrinsic factor, a completely separate job, something to do with anemia, we know it. HCl, what they do, they activate pepsinogen. Pepsinogen is secreted by chief cell. So in the exam, if I ask which is secreted by chief cell, answer is pepsinogen. Who activates pepsinogen? Which molecule which molecule is responsible activating pepsinogen? HCl. What pepsinogen active to? They become active as a pepsin. What is the function of pepsin? Pepsin digests proteins. Now HCl have other job as well. For example, they have very good immune system agent because they can kill like 99.99% bacteria virus right there. So once we eat, like, it contains a lot of bacteria virus, but as soon as you swallow, it touches the acid kill them. They're hostile for those. They create a really hostile environment. So before we talk about enteroendocrine cell, let's understand the function of HCl a little bit more. So they kill most of the microorganism. They help you denaturing the protein. Not only they activate the enzyme, they help denaturing. Remember, we learned that protein undergo multiple folding, primary, secondary, tertiary, and quaternary. Those folding makes them, so it's not a straight line. It's not like a polypeptide, it's like a ball, like multiple protein. The multiple folding allows them to create a three-dimensional structure. We need to denature them. We need to break those hydrogen bonds and other covalent bonds to make it denature. That's what HCl can do. HCl can also help with breaking down some cell walls and connective tissue in the meat. And finally, HCl activate pepsinogen, which produce pepsin, and pepsin can chemically digest protein. So proteins are nothing but polypeptide. When pepsin breaks them, it makes a small polypeptide chain, like dipeptide, tripeptide, tetrapeptide, like a small one. And then more enzyme comes and release them to free amino acid. Remember, when we eat protein, we absorb amino acid. We do not absorb protein. We break down protein to amino acid and we absorb amino acid. That's the function the HCl and pepsinogen trying to achieve. Now, what is the enteroendocrine cell? Enteroendocrine means those are the hormonal cells. Those are the endocrine cells in the stomach area. They secrete hormone. They secrete two important hormones, gastrin and somatostatin. Gastrin and somatostatin. So what gastrin does is gastrin is basically it just shows my Bluetooth is on. Just a second. Okay, enteroendocrine cells, two of them, two different class of cells, they secrete two different type of hormones. So one group is called G cells, G for gastrin. So they literally call G cells and gastrin, and D cells, D as in David, D cells for somatostatin. So G cells, when they secrete gastrin, what gastrin does, they activate these two cells. So the gastrin goes up and activating parietal and chip cells. So they're asking them, hey, secrete HCl, hey, secrete pepsinogen. So we have now protein in the system, we are going to digest them. So gastrin can be activated by the protein itself. So as soon as you eat protein, gastrin keep doing this. On the other hand, somatostatin is another control. So somatostatin, what they do, they inhibit gastrin production. So the D cells, they inhibit gastrin production. So at rest, we do not want this HCl to be produced because HCl is acid. We don't want. When we are not digesting anything, we're not eating protein, we don't want this to happen. So then somatostatin will inhibit at the gastrin at rest. But your food, like your nervous system, when you eat food, your nervous system can overrule that inhibition process. So they overrule the somatostatin inhibition process, and the gastrin keeps secreting. That's why sometimes this is a side note. Like sometimes if you eating too many times, like chewing gums. So in chewing gums, when you're chewing, you're not eating, meaning you're not eating any food, but your brain thinks that you're eating. So this mechanism is then the block. So that means somatostatin are not inhibiting gastrin. So some people, they may enhance the secretion of the hydrochloric acid when they keep eating, like keep chewing the chewing gum. So you have to be careful because normally if you read the level, it's chewing gum should be taken after the food, because chewing gum, was discovered for the practice to cleanliness, so like cleaning your mouth, not to as a food so that you don't feel hunger, because that has a negative effect as well. Okay. So that's the function of stomach. So stomach literally has tons of function. Then the material goes into the, we learned this, then the material, this is the secretion of the parietal cells. So parietal cells secrete the hydrogen ion and chloride ion separately from them into the lumen. So that's H plus and Cl minus together, that hydrochloric acid. They also secrete a intrinsic factor, which helps you to absorb the vitamin B12. So now this material that digested, partially digested protein, partially digested carbohydrate, remember carbohydrate digestion here, and those are all goes to the small intestines. But a little absorption may take place. Almost 100% absorption takes place in the small intestine, but a very little, particularly like a little bit of alcohol, drugs, they can be absorbed in the stomach, which is insignificant. So now the small intestine, this is like I would call it like a powerhouse of the digestive tract, because this is where all the other digestion and almost 100% absorption takes place. Remember, only two digestive activity happens here: partial digestion of carbohydrate, partial digestion of protein in the stomach. Apart from that, any other digestion, the complete digestion of lipid, the completion of all the carbohydrate digestion, completion of all the protein digestion takes place in the small intestine. So small intestine is pretty long compared to the large. That diameter is small. That's why they could take in a cadaver, they're like 20 feet long. So the duodenum is the first part. Then the part is called jejunum, and the last part is called ileum. So from the pyloric sphincter, between stomach and the duodenum. So from the pyloric sphincter, the small intestine goes all the way to a valve here called ileocecal valve. Ileo came from the word ileum. Cecal came from the word cecum. This area of the large intestine called cecum. We're going to cover that later on. So from here to here, so from pyloric sphincter all the way to ileocecal valve, that's basically our small intestine. So small intestine from the pyloric sphincter all the way to ileocecal sphincter or ileocecal valve. First part called duodenum, like almost like as I said, like 20 feet long. in a cadaver, but because of the muscle tone, it's like can fit in the abdomen easily. The lengthwise duodenum is the smallest, like less than a foot. Then close to 8 feet is the jejunum, and then more than like 11 feet is the ileum. So that's like a very long tube. Now, if it is a simple tube, like a simple rubber tube long, then it would have, if it would have the 20-feet tube with that diameter will have 3.6 square feet surface area. So if it is a pipeline, these are the surface area of the pipeline, inside surface area we are concerned about. So if you cut it open, if you measure the surface area of the small intestine, if it is a 20-feet pipeline with that diameter, it will be 3.6 square feet. But in reality, the surface area of small intestine is like close to 2,200 square feet. So from 3.6, which is like a small table, to 2,200 square feet, like a single family home. Like how they achieve that huge surface area. The small intestine undergo with all those four walls, mucosa, submucosa, muscularis externa, and serosa, they undergo a little bit anatomical differences. Like they make a lot of bumps. So this is what they do. They, first of all, not this one. First of all, they create plissé, like a fold in the small intestine. So if you look inside the small intestine, if this is the small intestine, if you look inside the small intestine, there are permanent fold they create. So whenever there is a fold, like ups and downs, obviously they will increase the surface area. So that's ups and downs increases the surface area. So, so this is inside of a small intestine. This inside of a small intestine, they created these folds, big folds, ups and downs, like this. So this fold increases the surface area. There are like approximately 800 folds. Those folds are called plissé. They're the intestinal permanent fold. Now, on top of this plissé, they have like a finger-like projection, like this, finger-like projection. So the entire small intestine. So now it's more, these finger-like projections are called villus is the singular one, the villi is the plural. Now, if you look at individual, so that one singular projection have columnar cells, simple column cells. So they are like, looks like this portion looks like a bristles on the brush. So those, the entire concept with the plissé, the permanent fold, 800 of them, and then all the villi, the finger-like projection, and within that, like bristles, like when you have a toothbrush, the bristles of the toothbrush, that's how it looks like, because of the columnar cells. So these are those microvilli. Those are called microvilli. So this is villi, the whole thing is villus is singular, multiple is villi. Then these are called individually microvillus, individual columnar cells, but multiple of them, we call them microvilli. But they looks like a brush, like when you have a toothbrush in the front of the bristles. So this is known as, top part is called brush border. So the enzyme is secreted by all these cells right here. There are a lot of goblet cells, and there are a lot of enteroendocrine cells. There are a lot of exocrine cells that exist in this area. Those are called brush border enzyme. That's a word. So when you see the word brush border enzyme, that means the enzyme secreted by small intestine, because their lining is known as brush border. The reason is they appear like a brush. And the base right here, so this is the huge villi. At the base of this is the villus. So huge villas are not here. So these two villi, that is known as a base. That base is called crypts of Lieberkahn. Should be a capital letter L. That's the name of a science. So they create that crypts. the crypts, it's called crypts of Libercon. Why these words are important? Because this is where these enzymes are secreted. This is where the exocrine cells are there. This is where the enzymes continuously being secreted. And also they have a lot of enteroendocrine gland cells, meaning those cells secrete hormones. Those cells secrete hormones. There are multiple types of hormones. I'm going to mention that 2 seconds later. So in other words, small intestine, they're 20 feet long, tiny tube, like a tiny diameter tube, that's why it's called mold. But inside, they have lots of bumps. Those permanent folds are called plicae, and then they have a finger-like projection called villi, and then columnar cell standing like a brush is called microvilli. Because of presence of the villi, microvilli, and the plicae, the surface area of the small intestine is like 2,200 square feet, huge. Why you need that? Because you need to digest all the food. So your completion of carbohydrate digestion, completion of protein digestion, and completion of lipid digestion takes place. Not only that, you absorb. So in that villi, all the blood capillary and the lymphatic capillary, both of them help in absorbing food material. Remember monomers like glucose, fructose, or amino acid, or fatty acid, all of them are absorbing right there. Then when they cannot absorb anything else, those are called unabsorbed food, or food that cannot be absorbed, or those indigestible food moves to the large intestine. The large intestine take care of the rest. Okay, so let's come back one more time and see what are the job they can achieve. This slide is a summary of what small intestine does. It's like a 2 pages material in your book. They have tons of glands. Some of them are exocrine glands, they're simply releasing mucus and the enzyme. Some of them are endocrine gland. Endocrine gland means they release hormone. So arrow up means stimulate, arrow down means inhibit. Okay, so they secrete close to 2 liters of liquid. Duodenal gland, remember duodenum, jejunum, and ileum. So in the duodenum area, there's 2 very important glands, mucus and Brunner's gland. So remember, pH 2 is the chyme over the stomach. So when that pH 2 material comes into the small intestine, first thing small intestine wants to do is to increase the pH, because pH 2 is dangerous. They don't have that protection. Small intestine does not have the protection from that acid. Their wall is going to be damaged. That's why more secretion can cause duodenal ulcer, because ulcer means they like literally damage the mucosa membrane. You don't want that. So what do you have to do? You have to raise the pH. How do you do it? You secrete a lot of buffer, which helps reduce the acidity. So they secrete a lot of alkaline buffer. On top of that, pancreas deliver more alkaline buffer. So these mucus, they secrete, they produce a lot of alkaline mucus. So they produce, reduce the acidity. And they also, the Brunner gland does the same thing. They also secrete a lot of mucus, reduce the acidity, and also secrete a hormone called urogastrone. What that urogastrone does, as soon as Brunner's gland receive the chyme, they get activated. They secrete urogastrone. Urogastrone goes to the stomach, telling stomach, hey, please stop working. You don't need to, because now I get the chyme. Now I will digest. You stop working. So that information is given by not only urogastrone, but some other hormone as well. So urogastrone is one of them. What are the other hormones? Secretin, cholecystokinin, CCK, gastric inhibitory peptide, GIP, and vasoactive intestinal peptide, VIP. Now, what this 4 hormone does, they have some similarities. So secretin, they increase the bile activity. So secretin, cholecystokinin, GIP, gastric inhibitory peptide, all 3 of them reduces the stomach activity. Remember, I said, once they receive the chyme, they can tell stomach, hey, you stop. Now, what else they do? Secretin increase the bile activity, CCK increase the pancreatic activity, and gastric inhibitory peptide insulin secretion from the pancreas as well. Now, bile is the one that's responsible for emulsification of fat. I'm going to mention that later on, because the liver is the one that produces bile. So when you study liver, we are going to go back to that bile. And on the last one, vasoactive, vaso means vessel. So vasoactive intestinal peptide, what they do, they dilate the capillary, because capillaries are now ready to absorb. So when the dilation of the capillary takes place, they can absorb fast. That's exactly what happened. So they reduce acid production in the stomach. They increase the pH of the chyme. They break down all the food material. They absorb the material. So small intestine has a lot of functions. Now they can achieve this with the help of 3 accessory organs right there: liver, gallbladder, pancreas. With the help of those 3 accessory organs, they can achieve this job. So let's learn what they do. So this is the intestinal juice. So the pancreas. Pancreas, you already know it because that is an endocrine gland as well. So pancreas, the first thing you need to know, this is a dual gland, meaning they have endocrine function as well as exocrine function. Endocrine function, we already learned, the islet of Langerhans, they secrete insulin and glucagon. Insulin reduces the blood sugar, meaning glucose goes to the cell. Glucagon increases the blood sugar. That's the endocrine system. Now, the remaining portion of the pancreas, those are called acinous cell, plural is acini. In the pancreas histology, this is like islet of Langerhans. These are islet of Langerhans. Any other cells that appear a little bit darker colored, those are called acinous cell. Plural is acini. What did they have? They have a cells with the duct. What they do, they release their secretion into the duct, main duct. So there are 2 major duct systems in the pancreas anatomically. This one is called main duct or main duct of the pancreas or pancreatic main duct. And that is a main branch. So accessory duct, main branch. So the main duct is not written on the slide. It's called duct of Wiersung, W-I-R-S-U-N-G. Duct of Wiersung is the name of the main pancreatic duct. And the accessory duct right here, the accessory pipeline is called duct of Santorini. Santorini, S-A-N-T-O-R-I-N-I, name of a scientist. So duct of Wiersung is the main duct. Duct of Santorini is the accessory duct. Both those ducts bring all the secretion that exocrine pancreatic acini cells release. So these are all exocrine secretions. They contain a lot of enzyme, a lot of buffers. Hormone is released by alpha and beta cells. That's in the islet. We learned that endocrine islet of Langerhans. Those are alpha and beta cells. Alpha cells secrete glucagon, beta cells secrete insulin, and we already know their function. This is what the pancreatic exocrine juice has. They have buffer, number 1, lots of bicarbonate buffer, and then they have carbohydrases, lipases, nucleases, proteases, meaning these are the ones they're breaking carbohydrate, lipid, nucleic acid, and proteins. We just discussed this. This is the buffer, the primarily bicarbonate buffer, neutralizes the acid that's coming from the stomach. We discussed that. These are the enzymes. So these, for example, some of the enzymes we got to memorize. So these include these. So enzymes, what do they include? Trypsinogen. So trypsinogen, again, trypsinogen, chymotrypsinogen, these are ogen, it's inactive. So they become activate in the small intestine and they become trypsin, chymotrypsin. So what they do, they break down proteins. Then you have carboxypeptidase. So they can break down peptide bonds, meaning they can break down protein. So these are protein breaking enzymes. These are all lipid and carbohydrate and nucleic acid breaking enzymes like amylase, lipase. Remember salivary amylase here? Pancreas also is called pancreatic amylase. So they can break down those same things. Next is the liver. Liver is the largest gland of the body, huge gland. They have tons of function. So first, the anatomy, 4 lobes, right lobe, left lobe, quadrate and quadrate lobe. So right lobe, humongous, right lobe, left lobe. If you turn over the other side, you have the quadrate lobe right here, quadrate, and the quadrate lobe. So liver, largest gland of the body, has 4 lobes, right, left, quadrate, quadrate. This this ligaments right there on the top of the liver. There are two major ligaments, right? There are many, but just focus on only two. That's actually only one is fine. This huge ligament right here in the middle between two lobes are called falciform ligament. That is keeping the left lobe and right lobe separate. And there's lots of cells that makes the liver called hepatocyte. It looks like a cycle with a spoke arrangement, okay? Now, what is the function of the liver? Liver has tons of functions, starting with like blood clotting to an immune system, tons of functions. Only one function we are interested right now, only one function. Liver produce bile. Liver produce bile. That bile is necessary for fat digestion because bile emulsify fat. Because fat is hydrophobic in nature, meaning they don't want to get mixed with the liquid of watery content. Enzymes is in the watery content. So if they're hydrophobic, enzymes in the watery content, they don't want to break down those. So what are you going to do? What we can do is we can emulsify the fat, meaning we can break the fat into a tiny droplet so that enzyme can attack them. So bile is a chemical to use in a physical breakdown of the fat. You can remember this way. So basically, if you take a 1 teaspoon oil in a glass of water, you will see that the oil is floating on the top. But now if you cover the glass and if you really shake, the oil will produce like tons of millions of bubble everywhere. That's what we do. So emulsifying fat so that enzymes can digest them efficiently. So fat emulsification is a necessary step for fat efficient digestion. That is why without bile, we cannot efficiently digest fat. Efficiently is the keyword. We cannot efficiently, and that is why although lingual lipase is producing here, the efficient digestion of fat we consider begins in the small intestine. So liver produces that bile. So liver produces that bile. You can read that from here. You can read this right there. The bile. So the yellowish green solutions contain mainly, they have a lot of bile pigments. The bile pigment, main thing, they come from heme. Remember bilirubin? So heme in the macrophage converted into biliverdin, and then the green color to bilirubin, and then it comes to bilirubin is one of the chief, the major bile pigment. So when liver is doing too much of them, and then the bilirubin become lots of blood, some kind of problem in the liver, so-called like jaundice, your body will turn like yellow because that bilirubin behaves like that. So that's basically the bile. So main function of bile is to emulsify fat. They also facilitate fat and cholesterol absorption. Okay. They help solubilize cholesterol as well. So this is a little bit that happen in the large intestine. This is how the bile is delivered, very important. Bile is coming from the, remember, right lobe of the liver and the left lobe of the liver here. So the underneath the right lobe, there is another organ called gallbladder. Underneath the right lobe of the liver. So if you go back right here, underneath the right lobe of the liver, that's a gallbladder. That sac is the gallbladder. That sac is the gallbladder. So what is the function of gallbladder? Gallbladder stores excess bile. So when the bile is yellow color, but when they lose a lot of water to store them condensed form, they become like a little bit greenish. That's why they appear greenish in this picture. So bile is produced by liver. Bile is stored, excess bile is stored by gallbladder. So gallbladder storing that bile. So right here, the bile, excess bile is stored. So one more time, the right hepatic ducts and the left hepatic duct, together they become common bile duct. Right hepatic ducts from the right lobe, left hepatic ducts from the left lobe of the liver, they become common bile duct. Then common bile duct is running. It's totally coming right here to the duodenum area. That's the pancreas. That's the duodenum area, the first part of the small intestine. So they use the same sphincter, the valve, where the pancreatic main duct is. So the main pancreatic duct sphincter, and then bile has a separate sphincter, and then together they have a one major sphincter. All the sphincters are controlled by those hormones like CCK, secretin, and thereby that's why they're controlling the sphincter, they control the availability of the bile solution in the duodenum area. And remember, bile is required for fat digestion. Because so as soon as the fat reaches here, the lipid molecule reaches here, they trigger this whole process. They trigger this whole process. So those hormone goes out, they open those valves, boom, bile comes. Now, when they have excess bile, they close those sphincter because fat digestion, let's say, is over, or you're not eating fat at that moment. So they close those sphincter, then the excess bile goes back with the same common bile duct. The excess bile goes back with the same common bile duct, and then finally goes through the cystic duct to the gallbladder. So the duct system you must remember. So right and left hepatic duct form a common hepatic duct. They merge with the cystic duct, form a common bile duct, bringing it here. But cystic duct take the solution from this direction. The common hepatic duct send the solution into that direction. So this is going towards pancreas, and when these are closed, they go back towards this, to the cystic duct, to the gallbladder. And gallbladder store the excess bile. The gallbladder stores the excess bile. So they store the excess bile, and they get rid of the water so they can store more bile. That is why sometimes, if the gallbladder doesn't properly function, they deposit stone. We call gallstone or gallbladder stone. Painful process when the stone get stuck into the cystic duct or any other duct system. So those are basically the three accessory organs: pancreas, liver, and gallbladder. All of them are dumping their secretion into the small intestine, and small intestine being a huge surface area, availability of all the enzymes, they not only complete the digestion, but also they absorb almost 100% nutrients there. So this is basically all about absorption and digestion of all these few slides. Then comes to the large intestine. So now the unusable food matter, the food matter that we didn't use it, contains lots of fibers or anything indigestible, move into the large intestine. So anatomically, large intestine, their diameter is more, that's why it's called large, and it has 5 regions: cecum, appendix, colon, rectum, and anal canal, or we can say like rectal canal and anus. So those are the 5 areas. So this is what the areas are. Like this is called appendix. It's like a worm-like structure. That's why it's called vermiform. It's worm-like structure, appendix. That's pouch, huge pouch is called cecum. Then from here, all the way here is the colon. This portion is rectum, and that's the anal canal, or you can call that part anus. So appendix, cecum, colon from here all the way up to here, rectal canal or rectum, and anus. Now, colon can be subdivided into 4 more regions. So colon, when they're going upwards, right here, we call ascending colon. When they're going from the right side to the left, we call transverse colon. And then when they're coming downwards, we call descending colon, coming downwards, descending colon. And then forming a structure like this called sigmoid colon, like a sigmoid colon. Finally, rectal canal or rectum and the anus. Now the colon, they create like a pouch-like structure for better absorption. So pouch-like structure, this pouch individually are called haustrum. And haustra, H-A-U-S-T-R-A, haustra is the plural of that haustrum. So this pouch-like structure, so these are haustras, all these are. Okay. So up, ascending, transverse, descending, and sigmoid colon. So that's the huge part. And the pouch-like structure right here is called cecum. So from the small intestine, all the unusable kind, the food matter, unusable food matter, they deliver right here through a valve. That valve here is called ileocecal valve, the valve between ileum of the small intestine and cecum of the large intestine. The valve is called ileocecal valve. So everything dumped right there. And then what large intestine does. So this is basically what the large intestine looks like. And then what they do, their main job is to compact those unusable food matter into feces. So they compact the feces by reabsorbing all the water. Because remember, body is adding tons of liquid. No matter how you drink, let's say 2 liters of liquid you drink a day. So 2 liters liquid you drink, a liter and a half is secreted by the saliva. So the stomach adds like another liter and a half. Then the small intestine add a liter. Bile is like another liter. So there's like almost like 8-9 liters of fluid available in the large intestine. That's why when large intestine cannot function properly, diarrhea, meaning lots of liquid going out, and that can deprive you from lots of salts. That's why when sodium, potassium, and bicarb deficiency occurs when you're large intestine. So large intestine is a great function because they reclaim water. You don't want to be dehydrated. So they reclaim water and they compact the feces. So that's their main job. What is their side job a little bit? Because they house, the colon house a lot of helpful bacteria. Those helpful bacteria give us, they leave us in our body, they live in our body synergistically, meaning they help us, we help them, mutual. So they give us vitamins, the vitamin K, biotin, biotin, some book will say vitamin B7, that's the name of biotin. In some country, they also call B8, like in France, biotin is known as B8. Anyway, the vitamin K, biotin, B5, all these vitamins come from large intestine. So those helpful bacteria in the colon helping us getting all these vitamins, because these vitamins have a tremendous role in your body. That is why when you take a very strong antibiotic, doctors will also recommend you, hey, take some yogurt, because yogurt has a lot of bacillus. They can grow back into the colon again. Obviously, they deals with organic waste like urobilinogen, starcobilinogen. Those are the fragment of the bile. Yeah. And they deals with a lot of toxins, so they can make sure they get rid of everything. So that's the function. So this particular slide is I found somewhere. It's like a summary of whatever we learned. This is the digestion. That look what happened to carbohydrate, what happened to lipid, what happened to protein. We're going to discuss that later on. I believe this is whatever all this digestion, and finally I think absorption. Water, ions, vitamins, they absorb directly, and these are all the liquid that we added, and we only get rid of like 150, 200 ml. That comes out with the feces. So think about like drinking 2,000, 1,500 saliva, gastric 1,500, so 3 + 2, 5, 6, 7, 8, 9. Yeah, so 9 liters of liquid and 9.2, and you're losing only, let's say, 0.15 or 0.2, so 9 liters liquid at the large intestine or the colon is basically reclaiming. So that is the major function of the large intestine. So when you study this entire chapter, you need to know first, what is the digestive tract look like? So mouth to pharynx to esophagus, stomach, small intestine, large intestine. In the mouth area, excuse me. In the mouth area, you have teeth, tongue, salivary glands. Those are the 3 accessory right here. In the abdomen, you have pancreas, gallbladder, liver. Those are the 3 accessory in the abdomen. So now when you study mouth, you should also study teeth, tongue, and salivary gland all together. Then study pharynx and esophagus. They're the transporters. They don't have any digestion or absorption functions. They transport the food to the stomach. Stomach has a lot of functions. They not only temporarily store the food, they denature, they help killing microorganisms, they denature the protein, they digest, partial digestion of protein takes place. A lot of hormonal function as well, along with the interesting structure. Then the food goes to the small intestine. Small intestine by far is the most things happen there, meaning lots of absorption, lots of digestion, and almost all the absorption happens. So they increase their surface area. So you should know the plicae, villi, microvilli, what is brush border. And when you're studying small intestine, the same time you can study the pancreas, liver, and gallbladder, because those are the three accessory organs helping small intestine to achieve all their function. Then anything unusable, anything that we could not digest, anything we could not absorb, that goes into the large intestine. Large intestine, they compact the feces by reclaiming almost 9 liters of water, and they get rid of the defecation, meaning they get rid of that unusable food matter. On top of that, they're the house of some helpful bacteria, which mutually help us. So it is basically a very long chapter, but it is not difficult.